Hello friends, welcome to my course on Microbial Biotechnology. We are in module number 11 discussing microbes in alternate energy. In today's lecture, we will be discussing about microbial fuel cells and microbial electrolysis cells. The lecture is broadly divided into three sections. In section 1, we start with an introduction to bioelectrochemical systems. Then we also briefly discuss about the historical development of bioelectrical systems, electrochemical systems, their significance, major advantages and current challenges. Then we discuss about chemical fuel cells and microbial fuel cells, then bioelectrochemical technologies, MFCs and MECs. Then we make a comparison between the two. Then we will discuss about microbial fuel cells in detail, particularly the concept of MFCs, design and working principles, electron transfer mechanism to anode and then cathode. Then factors affecting the electron transfer mechanism, the mechanisms of biofilm formation in MFCs, anode and cathode material selection, anode electrode materials using natural waste resources for MFCs. We will discuss about the details of the other two sections as we come across them. So, what are bioelectrochemical systems? These are unique systems capable of converting the chemical energy of organic waste, including low strand wastewaters and lignocellulosic biomass, into electricity or hydrogen chemical products in microbial fuel cells or microbial electrolysis cells respectively, or other products formed at the cathode by an electrochemical reduction process. So, basically this BES comprise of two things, microbial fuel cells and microbial electrolysis cells. So, how and when these bioelectrochemical systems came into being? The concept of microbial electrochemical systems like MES and MFCs dates back to 1911, when Michael C. Porter demonstrated that electricity can be generated from microbial metabolism. In the 1970s, MFC research gained momentum with studies on mediator based electron transfer. Then in the early 2000, there were a lot of advancements, particularly with GeoVector and Swayam highlighted direct electron transfer mechanisms, sparking interest in practical applications. Microbial electrolysis cells emerged in the mid 2000s as an offshoot of MFCs, which focused on hydrogen gas production by applying external voltage. Now, what are the significance of these bioelectrochemical systems? They help us in sustainable energy production. BES offer renewable and eco-friendly solutions for electricity and hydrogen gas generation. They help us in waste management. MFCs and MECs transform organic waste into energy, aiding in wastewater treatment. So, this is basically a method by which we can convert waste into wealth. It has a lot of bioengineering potential. They provide a platform to study and optimize microbial electron transfer, enabling advancements in microbial biotechnology, and it gives us a lot of environmental benefits. For example, BES reduce reliance on fossil fuels and contribute to a circular economy as well as a green economy. They have multifunctionality. BES can simultaneously treat waste, produce bioenergy and act as biosensors, making them versatile tools in biotechnology and environmental sciences. So, these systems have a lot of advantages, and also they suffer from certain challenges. So, like for example, we have the scope of obtaining green energy by these BES. But while offering so many advantages, which is discussed in the earlier slides, they also suffer from certain disadvantages. So, some of the disadvantages are related to technical issues. For example, it provides lower power output and slow reaction rate, although BES helps us in obtaining electrical energy from waste, but the power output is actually very low and the reaction rate is very slow. And another problem is the substrate specificity and biofilm management. These biofilms are very important in the production of electricity, and we will discuss about them in detail later. So, managing these biofilms is actually a challenge. Then there is a constraint of materials. For example, the electrodes are very expensive and the membranes degrading. So, therefore, make the system a little bit very costly. Then the challenge is again there from the biological factors. For example, microbial maintenance is an important issue, because without the presence of the microbes we cannot carry out the metabolic reactions which help us in generating the electricity. So, their maintenance is a challenge, and the sensitivity to environmental changes and the risk of contamination is also one of the important challenges. Another issue is the scaling and commercialization. So, in a small scale viable model, it is possible to obtain say some power output, but we want to scale it up, the system becomes very complex. So, there are economic challenges, and also it lacks standardization, and then sustainability concern is also there, resource demand, and then waste management and limited system durability are some of the challenges of BES. Now, let us have a peep into the chemical fuel cells and the microbial fuel cells. So, here you can see that chemical fuel cells use chemical fuels like hydrogen, here you can see, or methanol with precious metal catalyst, example platinum, to generate energy, often requiring controlled conditions like high temperature and pressure. They are efficient and produce water, as you can see over here, as byproducts, but may rely on non-renewable sources. So, the energy fuel that is coming into this system is actually maybe from non-renewable sources. In the case of microbial fuel cells, you can see a similar design with an anode and a cathode, where water is produced, and then there is a membrane which separates the anodic and the cathodic chamber, and there is a transport of the ions or electrons from one chamber to the other. So, here you can see, instead of the fuel which is non-renewable, we may have organic waste or organic materials as a fuel. So, microbial fuel cells are BES that harness microorganisms act as biocatalysts to convert organic matter. On these anode you can see some rod shaped microbes given as a example here in this illustration. So, these microorganisms act as biocatalysts to convert these organic matter such as wastewater or biomass into electricity under ambient conditions compared to high temperature and pressure conditions. So, while these are less efficient than the CFCs, MFCs are eco-friendly, offering the dual benefit of bioelectricity production and waste treatment. So, what are the important bioelectrochemical technologies? Number one is the MFCs and the MECs. These microbial fuel cells are bioelectrochemical systems in which microorganisms actively participate in direct conversion of chemical energy embedded within organic materials into sustainable electrical energy. They are primarily utilized for bioelectricity production, being economically feasible and an eco-friendly approach. MFCs provide immense potential as an alternative to the uncontrolled use of fossil fuels. Microbial electrochemical cells refer to a device where microorganisms play a crucial role in catalyzing electrochemical reactions, typically involving the oxidation of organic substrates at the anode and reduction of electron acceptors in the cathode. MECs are designed to harness microbial metabolic activity to generate electrical current or produce valuable products, making them a key component in sustainable energy. Let us now have a comparison between the MFCs and the MECs. So, here you can see a diagram where in the anode you can see the organic substrates are getting converted into energy, and then here the anode is all lined up or layered with microbes, and there is a elective membrane in both the cases. Now, let us look into the differences or similarities between them. So, if you look into the primary output of MFCs, it is electricity, but in the case of microbial electrolysis cells, it is hydrogen gas, you can see over here. And then the key reaction in MFCs are electron flow from organic matter metabolism powers an external circuit, you can see over here. And in the case of the MECs, electrochemical reactions produce hydrogen at the cathode. The application of MFCs are in the case of wastewater treatment as sensors, remote power generation. In the case of MFCs, it is renewable energy storage, biohydrogen production, and wastewater treatment. In the cathode, in the case of MFCs, oxygen reduction takes place producing water. In the case of MECs, it is proton reduction which leads to the production of hydrogen gas. Regarding the voltage requirement, no external voltage is required in the case of MFCs. Microbial metabolism drives the reaction. In the case of MECs, external voltage is applied to enhance hydrogen production. So, this is in brief a comparison between the MFCs and MECs. So, these microbial fuel cells is having huge potential as a green energy supply of wastewater treatment plants. So, here in the case of an MFC device, the input is influent or waste water, which leads to the generation of bioelectricity while producing sludge, which is actually further used for landfilling or conversion into some other products, and the effluent is clean water. So, we can convert wastewater, treat wastewater by the help of an MFC device in producing clean water, and in the process generate bioelectricity, and also some material, sludge material, which can have further use in other applications. So, what is the concept of MFCs? Electroactive microorganisms are crucial for the power output of microbial fuel cells and have diverse applications in energy production, wastewater treatment, biosensing, water distillation, and chemical production. The two main types of electroconductive microbes that are used in bioelectrochemical systems are exoelectrogenic microorganisms. These microorganisms, typically bacteria, transfer electrons to insoluble electron acceptors such as solid electrodes, oxidizing organic compounds to release electrons, which are then used for electricity generation in microbial fuel cells. Then we have electrotrophic microorganisms. These are microbes which pull electrons from the cathode either directly or indirectly, serving as electron acceptors and offering an alternative to oxygen in cathode reactions. Let us now look into the design and working principles of MFCs. As a biological technology, MFCs integrate cathode and anode materials with microorganisms to achieve electricity generation, which we call as bioelectricity. MFCs utilize a strategy that includes the microbial conversion of chemical energy of organic and inorganic compounds into electricity. Basically, MFCs require 5 components. As you can see in this diagram, one by one we will discuss. An anodic electrode. This is the anodic electrode, within which electrons, protons, and other metabolites are produced through anaerobic oxidation of substrates. Then we have a cathodic electrode, which is required for collecting electrons by an electron acceptor material. Then we have a membrane. As you can see, this membrane helps in physical separation of the two anodic and cathodic chambers and helps in the mediation of the proton diffusion to the cathode from the anodic chamber. Then we have an external circuit which allow electron flow from anode to the cathode. And one of the most important component is the microorganisms which inhibit the surface of the electrode, particularly the anode. So, in the anode chamber, microorganisms, fifth important component of MFCs discussed in the earlier slide, particularly exoelectrogens inhibit the external electrode. Exoelectrogens metabolize organic matter, releasing electrons in the process. These electrons are then transferred to the anode, typically constructed from conductive materials such as graphite or carbon. These materials facilitate electron transfer and promote bacterial adhesion, forming a biofilm. So, you can see in the anode the reactions taking place, whereby the organic substrate is converted into hydrogen ions and electrons are produced, and carbon dioxide gas is also released. And here is an example given with a balanced equation. So, these electron produced are flowing through the external circuit to the cathode, and these protons or the hydrogen ions which are produced will diffuse through this elective membrane to the cathodic chamber. So, in the cathode chamber, primary role is to receive the electrons released from the anode. It contains an electron acceptor molecule such as oxygen, ferricyanide, mercury, iron, copper or chromium, along with a cathode electrode composed of porous material and a catalyst like platinum. The cathode facilitates oxygen reduction from the surrounding environment, where the reduction reaction combines electrons and protons to produce water. So, this is the reaction that is taking place here. So, we are converting organic substrates finally into water, which is a green product and not harmful to the environment. So, here the external circuit connects the anode and cathode, typically composed of copper or titanium wires. These circuit enables the movement of electrons released by bacteria from the anode to the cathode electrode during the metabolic process. This electron flow generates an electric current which can be harnessed to power various electrical devices. The only problem is that this power output is quite low. So, then there is a proton exchange membrane or the PEM, which we generally refer to as the elective membrane. This separates the anode and the cathodic chambers, as we have already mentioned. This membrane is crucial for facilitating the transfer of protons generated during bacterial metabolism from the anodic side to the cathodic chamber, while preventing the direct contact between the two electrodes. Commonly used membranes for this purpose include Nephion. So, what is the mechanism for delivering electrons to an anode? So, there is a direct electron transfer and MET, which are known as the major mechanisms for delivering electrons to an anode. So, here we can see a bacteria and which goes for the formation of a biofilm, and in these direct electron transfer exoelectrogenic bacteria, C-type cytochromes and pili, which we call as the nanowires, are involved in direct electron transfer to a solid state electron acceptor. So, from these electron is getting transferred through these nanowires or the pili. In the mediated electron transfer or indirect electron transfer, here electron transfer happens indirectly through an electron mediator. So, you have a mediator here which becomes reduced and then again oxidized in the process. So, these reduced mediator or the reduced electron mediator and then gets converted back to the oxidized electron mediator, and then this cycle keeps going on, and in the process it transfers the electrons to the anode. So, let us discuss about the direct electron transfer and mediated electron transfer, which are key to MFC performance. MET is often mediated by electron shuttles. Example, in the case of Pseudomonas aeruginosa, then Savanella oneidensis and E. coli, and this is actually a major limiting factor. The outer membrane of gram-negative bacteria hinders shuttle transport, reducing MET efficiency and MFC power. Enhancing membrane permeability, especially in gram-negative bacteria, can improve mediated electron transfer. Then we have the gram-positive bacteria with simpler compositions and higher surface zeta potential. They may have different electrogenic properties. However, differentiating between MET and DAT is challenging due to microbial metabolic diversity in the MFCs. The direct electron transfer involves certain microorganisms known as exoelectrogens transferring electrons directly from organic matter oxidation to the anode. This process requires physical contact between the microbial cell membrane and the electrode that occurs via outer membrane cytochromes, conductive pili or self-assembled attachments. So, here you can see the conductive pili, and the electron is getting directly transferred into the anode from these electron pili. Bacteria like Geobacter species and Savanella and Rhodophyrax are efficient in direct electron transfer. Geobacter sulfur reducens with its fully sequenced genome and 110 genes encoding C-type cytochromes is extensively studied for its role in extracellular electron transfer. Then in the case of fungi cells, we can see the presence of an redox enzyme which helps in the transferring of these electron into the anode. So, biofilm formation is essential for electron transfer in direct electron transfer, as it allows microorganisms to interact with the anode. C-type cytochromes have a redox potential range of around 1 volt and mediate direct electron transfer in the first layer of biofilm bacteria. Bacteria like Savanella and Geobacter species form electrically conductive nanowires for this electron transfer. Then there are monolayer biofilms which concentrate the majority of cells near the electrodes, while multilayer biofilms employ long range electron transport mechanisms like nanowires due to limited direct electrode access for the cells. Dense pili networks exhibit metal-like conductivity, significantly contributing to current generation, and are conductive pili produced by microorganisms like Geobacter, sulfur reducens, aiding electron transport between biofilm cells and the bioelectrodes. In mixed cultures, exoelectrogens engage in direct interspecies electron transfer using pili and pilus associated C-type cytochrome. In mediated electron transfer or indirect electron transfer, MET serves as an alternative electron transfer mechanism when microorganisms are unable to interact with the anode surface directly. So, here we do not see the presence of any pili. So, here there are certain mediators or electron shuttles. MET utilizes the redox carriers as electron transporters which gather electrons from microbial cells in their reduced state and transfer them to the anode material, subsequently oxidizing in microbial fuel cells. Basically, these electrons are shuttles collect the electron from these bacteria and transfer to the anode surface. These redox mediators can be categorized as exogenous, externally added, or endogenous, which are produced by the microorganisms themselves sometimes. Soluble mediators facilitate microbial electron transfer, including primary and secondary metabolites secreted by bacterial activity or artificially added mediators. So, here higher electrode redox potential mediators, example benzoquinone, potassium ferricyanide, enhance the power generation. Then there are synthetic mediators like metalloorganics and dyes, example methylene blue, which are effective but limited by instability and toxicity. Then we have the endogenous mediators. Microbes produce mediators like flavins, quinones, and piocyanins, for example, in the case of Pseudomonas aeruginosa, which aid in the electron transfer. Both gram negative and gram positive bacteria use endogenous mediators with mixed cultures, improving transfer efficiency. Despite thick cell walls, gram positive bacteria contribute to indirect electron transfer and electron transport in the MFCs. Now, let us look into the qualities of good mediators. Number one, it should have movability through microbial surfaces. Then it should be non-toxic to the microorganisms. It should be soluble in anolyte solution. Then it should be affordable. It should be able to produce sufficient redox potential for electron transfer. So, some of the examples of good mediators are phenazine, which are as the called as the self mediators, or you have 9,10 anthraquinone 2,7-disulfonic acid and piocyanin. Then we have exogenous mediators like methylene blue, riboflavin, humic acid, neutral red and thionin, or 9,10 anthracenedione. So, let us have a look into the mechanism of electron transfer in the cathodes. Cathodic electron transfer mechanism in microbial fuel cells have received less attention compared to anodic mechanisms. So, here we have these organic substrates which get converted into water. So, two types of electron transfers from cathode material to bacterial cells have been identified. Direct electron transfer here in this case from the electron to the microbe, it is getting transferred, and the indirect electron transfer where there are again electron shuttles. So, this is maybe happening through pilus or direct surface contact. And in the case of fungal cells, we may have certain biomolecules which mediate. So, direct electron transfer occurs when microbial cells directly contact the cathode material facilitated by C-type cytochromes in the external membrane of the microbial cell. A formation of a gradient proton or proton gradient during cathode oxidation is recognized as an important factor in cathodic electron transfer. So, indirect electron transfer involves soluble redox transporters where both cathode material and microbes reduce these transporters. Some studies have focused on cathodic MFCs where microbes inoculated in the cathode compartment for electricity generation. Certain bacteria like Acinetobacter secrete odd redox compounds with quinine-like properties facilitating electron transfer towards microorganisms. Biocathodes have been used to initiate oxygen reduction reactions to enhance electricity production. In rare cases, electron transfer from cathode material to microorganisms occurs through hydrogen oxidation. Then we have cathodic electron transfer, which plays a significant role in bioremediation for treating environmental contaminants and in the production of methane, carbon compounds and hydrogen in biocathodic microbial fuel cells. Let us now discuss about the factors which affect the electron transfer mechanism. So, number one is the biofilm integrity. The biofilm affects the diffusion of substrate molecules towards the anode, subsequently affecting the electron transfer mechanisms. Generally, the electrocatalytic function of a biofilm is affected by the source, nature and type of inoculum, influent concentration, operating conditions and structure and composition of electrodes. So, the electrode configuration structure has a pivotal role in microbial metabolism and electron transfer across the biofilm and electrode matrix. Common ones are the flat, packed, stuffed and brush models. However, the surface roughness of the electrode is involved in electron propagation. Then another important factor is the electrolyte and electron mediators. The high concentrated influent increases the acetogenesis and methanogenesis mechanisms which impair the electrolytic performance. However, the high concentration of mediators can lead to impaired electrodes because of toxicity and instability. Let us examine the first factor, the biofilm, and how these biofilms are formed in MFCs. So, biofilm formation includes a cascade of 5 stages. Number one is the motile microbial cells adhere to the surface in the first phase. Our first stage, as you can see here, fungal cell have adhered here, or the bacterial cells has been transported over here, and they adhere into this surface. The bacterial cells then aggregate to form microcolonies and begin excreting exopolysaccharides or EPS. Cell to cell adhesion results in the formation of multi-layered clusters forming a biofilm in the next stage, and the biofilm then matures and develops within the EPS matrix, protecting against environmental threats. Upon reaching a critical mass, the biofilm disperses cells which colonize other surfaces. Now, the anode and the cathode are the important components of a microbial fuel cell. Now, how do we select the right material for making the anode and the cathode? So, in the case of anode, carbon based materials, example felt, mesh, graphite, are commonly used for their conductivity and biocompatibility, because the microbes has to grow on the anodes and they have to support the growth of microbes. The anode supports microbial oxidation of organic compounds, releasing electrons and protons for electricity generation. Now, in the case of cathode, typically we use noble metals like platinum or carbon based materials with catalyst. The cathode facilitates reduction reactions, example oxygen reduction, consuming electrons and protons to complete the circuit and produce water or reduced products. We can also make anodes by using materials obtained from natural waste. For example, these are a list of commonly used electrodes. As you can see from A, this is carbon paper, then we have carbon cloth, then we have carbon fiber, and then we have reticulated vitrified carbon, then we have a carbon mesh, then we have granular graphite which is in the granular form in F, then we have the carbon brush in G, and then we have graphite rods, as you can see over here, or we may have polycrystalline graphite as shown in I. Then we have carbon felt in the case of J, and then platinum mesh also can be used. Then different metal electrode strips can be seen over here with in these pictures, like silver, aluminum, copper, nickel, zinc, iron, and then we have the conductive polymer based strips which are non metallic. So, there are the various anode materials listed in these tables, starting from CANF to compressed milling residue, bamboo charcoal, then we can use loofah sponge, and then coconut shell, and then sewage sludge, and then barbed chestnut shell and silk cocoon, and also chestnut cells and onion peels, coffee waste and so on. And then the surface area of these electrodes are given, and also the dimensions of these electrodes or the size of these electrodes are given, and the inoculum source, the microbe population is obtained, for example, in the case of CANF from domestic sewage, or we may have anaerobic mixed sludge in many cases, and then we have mixed sludge in certain cases, and then also mostly coffee waste is kind of domestic waste. And we can see that here is the power density milliwatt per square meter. In certain cases it can be very, very high, as in the case of coffee domestic waste. Let us now go to section 2, where we will discuss about the various types of microbes or the microbial communities which are used in the MFCs, particularly their diversity and the function. And we will also discuss about the pure culture versus mixed culture in these MFCs, and what are the types of MFCs. Let us begin with the microbial community, which may come from bacteria, algae, or fungi. We may have gram negative bacteria like facultative anaerobes, Escherichia species, Swanella, Shigella, Salmonella, and we have anaerobes like Geobacter, D-sulfovibrio, then D-sulfuromonas, and then we have aerobes like Pseudomonas, Acinetobacter, etc. Then in the case of gram-positive bacteria, we may have facultative anaerobes like Staphylococcus, Streptococci, Enterococcus, Mycobacterium, Bacillus, and Corynebacterium. And in the case of anaerobes, we have Clostridium, Actinomyces, and so on. In the case of algae, we have the Chlorella, and then we have Nanochloropsis, then we have Dunaliella, and then Hematococcus, Desmus, and Spirulina, and then we have Syncoccus and Chlamydomonas species, etc. Amongst fungi, we can have Coriolus, Saccharomyces, Agaricus, Trichoderma, and so on. Now, let us make a comparison of bioelectricity generation using pure culture versus the mixed culture. So, in the case of electron transfer efficiency, in pure culture we focus studies on specific microbes, and in the case of mixed cultures we look into the synergistic interactions which enhance the bioenergy production. From the substrate utilization point of view, we require selective substrates and precise operating conditions for pure cultures. However, mixed cultures utilize a broader range of substrates, increasing the efficiency. And then for electron activity, in case of pure culture, certain species like Geobacter may exhibit very high activity. In the case of mixed culture, diverse microbes collectively improve overall electron activity. Practicality for large scale applications point of view, pure culture is limited by cost, substrate specificity, and contamination risk. In the case of mixed cultures, it is a more practical approach with lower substrate and contamination concerns. In the case of bioaugmentation of the potential, pure cultures have improved startup time and performance in controlled conditions. In mixed culture, diverse metabolic capabilities and naturally enhance the performance. Now, what are the different type of MFCs? Microorganisms based biological fuel cells include photosynthetic MFCs or PMFCs, then microbial desalination cells or MDCs, and then sediment microbial fuel cells or SMFCs. So, these photosynthetic microbial fuel cells convert sunlight into electricity through metabolic reactions. Innovative technologies based on a combination of sunlight and microbial power have been an effective strategy for improved voltage generation. So, here this is a schematics of a photosynthetic MFC. So, sunlight is the source of the energy over here, and you can see in this anode different types of photosynthetic organisms are allowed to grow, which break down water into protons, which are transported across the elective membrane into the cathode, where with the incoming electron which was produced in the anode and transferred through the external circuit are utilized in the conversion of oxygen into water. So, several microalgae, cyanobacteria, and plant species have exhibited remarkable capacity to optimize voltage generation in these photosynthetic microbial fuel cells. Let us now discuss about the microbial desalination cells or the MDCs. Conventional destination plants are energy intensive and consume roughly around 3.7 to 650 kilowatt hour energy per cubic meter of water desalination. MDCs are important for simultaneous organic waste and wastewater treatment, water desalination and electricity generation through the oxidation of the organics. So, here is a schematic of microbial desalination cell which is adapted from Cow et al. 2019. MDCs are specialized MFCs with a third chamber for water desalination. Ion exchange membranes AEM and CEM. So, here you can see these membranes AEM and CEM. On either side of the saline filled middle chamber directs anions to the anode and cations to the cathode. So, here we have the saline filled water in this middle chamber. So, the anodic biofilm oxidizes organic matter here you can see driving electron flow and ion migration enabling desalination up to 99% and power generation through the electrode potential difference. In the case of sediment microbial fuel cells, this technique enables simultaneous heavy metal bioremediation, bioenergy reproduction and wastewater decontamination. As a membrane free system, here there is no any membrane as you can see, electrons flow through an external circuit while water facilitates the proton transport. In a simple sediment microbial fuel cell, the anode is buried in anaerobic sediment containing exoelectrogens and the cathode is placed in the overlying water as you can see in this diagram. Based on the setup of the chambers, MFCs can be classified as follows. They may be single chambered or they may be double chambered, and there are another type upflow MFCs and they are stacked MFCs or layer by layer 3D miniature MFCs. Let us first look into the single chambered MFCs. These single chamber MFCs feature a simple cost effective design with a single chamber housing both the anode and cathode. A porous cathode facilitates proton movement often without a proton exchange membrane. So, here you can see this is the anode chamber where the microbe has grown on the anode and the biofilm is formed and it catalyzes the conversion of waste and then electrons are released which flow through the external circuit which is connected to a cathode which is exposed to air. So, also called as the air cathode. So, this may be a porous or it may be and it may be not having a proton exchange membrane. These schematics is self-explanatory. It shows how simple this system is. So, there are some variations which include membrane MFCs with a PAM between the anode and the cathode and atmospheric oxygen supports the cathodic reactions. Here we focus on improving bioenergy outputs, especially scalability and integration with wastewater treatments. Then in the case of double chambered MFCs, two chambers are available here which are basically the anodic and the cathodic compartment which is separated by a PAM or a salt bridge. The anode hosts the bacterial oxidation while the PAM transfers protons and limits oxygen penetration. These are operated in batch or fed batch modes. It suits autonomous sensors and wastewater treatment. Despite high pumping energy requirements, it offers advantages like controlled cathodic action, improved flow rates and mediator addition for efficiency enhancement. So, there are many other types of MFCs. Number one is the A here is the upflow MFC, stacked MFC in the case of B you can see, and the layer by layer 3D miniature MFC. So, due to shortage of time we will not be able to discuss all of them in detail. Let us now move on to section 3 where we will be discussing on microbial electrolysis cells, then their working principles and what are the materials suitable for anode and cathode. In the case of MECs, what are the different applications of them, carbon dioxide reduction with MECs, particularly carbon sequestration, methanolins and their inhibition to hydrogenyl. We will also discuss about synthesis mechanisms of value-added bioproducts on MEC cathode surface, Then we discuss about the critical factors affecting MFC and MEC performance, and then economic and environmental assessment of MFCs and MECs, and future prospects of bioelectrochemical systems. Microbial electrochemical cells, particularly their design mainly consists of two parts, an anode chamber and a cathode chamber, similar to the earlier case. These are separated from each other with an ion exchange membrane. However, different configurations of MECs have been developed over the past few years for the improvement of hydrogen yields. So, this is the double chambered microbial electrochemical cell with a membrane, and this is a single chambered microbial electrochemical cell without a membrane. So, in the double chambered MEC, we have certain advantages, like the membrane prevents back diffusion of hydrogen from cathode to anode, and it maintains the purity of the hydrogen gas. However, it has certain disadvantages. Large distance between the electrodes increases the system resistance, which leads to substantial increase in the pH gradient, thereby resulting in potential losses. This increases the overall cost of hydrogen production. In the case of single chambered MECs, which are membraneless, we have advantages like the reduction in potential losses, higher current densities, and hydrogen production rates are faster, and the effect of pH gradients are also reduced. However, it has certain disadvantages like the production of methane by hydrogenotrophic bacteria, which decreases the hydrogen purity. Let us look into the working principles of the MECs. Microorganisms, exoelectrogens in the anode chamber oxidize organic substrates, example wastewater, biomass releasing electrons, protons, and carbon dioxide. Electrons released by microbes travel through an external circuit from the anode to the cathode, creating an electric current. Protons produced at the anode migrate toward the cathode through a proton exchange membrane or electrolyte. An external voltage is applied to the system to drive the reduction reaction. At the cathode, protons combine with electrons to produce hydrogen gas or other reduced products depending on the system design. So, you can see here the reactions at the anode and the reactions at the cathode, and the overall reactions where ultimately we have hydrogen production. So, various applications of MECs are there. Number one is hydrogen production, which we could see in the reaction. Then it helps in the case of sustainable agriculture, it helps us in bioremediation, it helps us in carbon dioxide fixation or carbon sequestration, and we can have energy production and storage and wastewater treatment. The primary application of MEC is the production of hydrogen gas, a sustainable and renewable method for generating hydrogen by anaerobic oxidation of organic matter. It also helps in the removal of contaminants and contributes to energy recovery from the waste, and thereby converting the waste into wealth. The hydrogen that is generated from this process can be stored and later used for various applications, including power generation through fuel cells or as a feedstock for chemical processes. By using organic carbon sources, MECs facilitate the capture of carbon in the form of hydrogen and prevent the release of greenhouse gases. MECs have the potential for bioremediation applications by the degradation of organic pollutants in contaminated environments, and these technology can facilitate the conversion of organic waste such as crop residue into hydrogen, while simultaneously treating the waste. Let us now discuss a bit about the carbon dioxide reduction with MECs and how carbon is sequestrated. Microbial electrochemical systems rely on microbial metabolism for functions like organic pollutant degradation, carbon dioxide capture, electricity production, and biochemical synthesis. As a subclass of MECs, MES focuses on cathodic carbon dioxide conversion using electrons and protons generated at the anode to drive electrosynthesis reactions at the cathode. So, here you can see direct electron transfer in the cathode, and indirect electron transfer in the cathode. Then the activity of methanogens such as methanobacterials, methanomicrobials, methanosarcinaceae and methanocetaceae have been shown to induce the opposite effect and suppress the hydrogen yield in the MECs. So, methanogens inhibit hydrogen yields. In fully anaerobic MEC operation, the functions of acetoclastic methanogens and hydrogenotropic methanogens have been exclusively studied to improve cathodic hydrogen recovery. In the case of acetoclastic methanogenesis, you can see the conversion of acetic acid to methane and carbon dioxide, and in the case of hydrogenotropic methanogenesis, these hydrogen and carbon dioxide combine to form methane and water. Methanogenesis management involves lowering the electrolyte pH, lowering the operating temperature and optimizing the applied voltage and low hydraulic retention time. Both acetoclastic hydrogenotropic methanogens promote the production of methane directly from acetate or after hydrogen production, thus reducing hydrogen yields. Production of hydrogen by mixed cultures associated with methanogens can lead to a higher cost of hydrogen purification. This is why pure cultures are used for MEC targeting H2 production. So, here these are the exo electrogens, and then we have these organic waste. So, these are directly metabolized by the exo electrogens in the anode, which you are now well conversed with, and then these also helps in the cathodic hydrogen evolution reaction. Then we have in certain cases fermentative bacteria, so which actually converts this organic waste into acetate, and then these acetoclastic methanogens convert it back to methane. And then again the hydrogen that is produced with cathodic reaction by this process may actually be converted back to methane by hydrogenotropes. So, this shows that the hydrogen yield is reduced due to this kind of reactions taking place, or even in case of certain other homoacetogens, this hydrogen is converted back to acetate, and of course, with acetogens it converted back into hydrogen, and of course, there are hydrogen producing bacteria which will directly produce the hydrogen from this organic waste and their metabolites. So, in a way these methane producing organisms, it reduce the yield of hydrogen, as you can see clearly in this diagram. What are the synthesis mechanism for value added bioproducts on MEC cathode surface? The microbial communities have the capacity for bioelectrochemical transformation of carbon dioxide into typical value added bio based products. Such products can be classified as C1, C2, C3 and C4 biochemicals based on a number of carbon atoms they contain. So, here we have these cathode, and then we have many microorganisms in this cathode. So, here they convert carbon dioxide and oxygen, combine them and may produce formate, and or they may produce methane, and this formate enters the transforms into acetyl coenzyme A, which will further produce ethanol, acetate. Then it may, the acetate will produce butyrate, that goes on to produce caproate and butanol, and caproate is converted to hexanol. Similarly, this acetyl coenzyme A gets converted to pyruvate and then fumarate, or pyruvate to lactate, and again fumarate to succinate, or the acetyl coenzyme A may directly produce propionate, which produces propanol. So, we can see that many products like propanol, lactate, succinate, hexanol, butanol, ethanol and are produced, which can be value added products, and this methane can be converted into methanol and finally formaldehyde. So, some of the critical factors which affect MFC and MEC performance are the system architecture, the operative parameters, techno-economic aspects and the biological parameters. The system architecture basically involves the cathode chamber, anode chamber and the type of membrane. Then in the case of biological parameters, whether it is a pure culture or a mixed culture, what is the source of the inoculum and the bacterial metabolic losses, the community dynamics play a critical role. Let us look into the economic feasibility of MFCs and MECs, or the evaluation of material construction and maintenance cost for both MFCs and MEC are important. Analyzing potential revenue or cost saving from electricity generation in MFCs and hydrogen production in MFCs are also important. Assessing the economic feasibility for specific applications like wastewater treatment or power generation for both the technologies are also important. Comparing the economic viability of electricity generation in MFCs and hydrogen production in MECs with other existing methods are also important while comparing the economic feasibility. Then we also need to look into the environmental assessment of the MFCs and MECs, conducting a life cycle assessment for both MFCs and MECs, considering a raw material extraction, manufacturing operation and end of life disposal is important. Assessing the reduction in greenhouse gas emissions compared to traditional energy or wastewater treatment methods for both the technologies, and examining environmental benefits and drawbacks associated with the applications of both MFCs and MECs. Evaluating potential environmental challenges or risks associated with the deployment of both the technologies are equally important in the environmental assessment. Now, let us look into future prospects of bioelectrochemical systems. We may use them for energy harvesting, enhance BES efficiency for energy recovery from waste, paired with advanced storage for reliability. In the case of bioremediation, we need to develop sustainable BES based solutions for contaminant removal in water and soil. Then in the case of sensor irrigation, we have to look for combined BES with sensor for real time environmental monitoring and adaptive management. We may use synthetic biology to create microbial communities with improved bioprocess and energy generation capabilities, or we may use hybrid systems where we integrate BES with renewables, example solar, wind, for versatile energy solution and optimize BES for high value bioproducts. So, with this we come to end of today's lecture. Thank you for your kind attention.